So yeah, hi, I'm Loren. Thank you for the introduction. So I was I was the first Melon Scholar. So yeah, it's really exciting to actually finally be here to talk about what I've been doing for the past 4 years, because I've come to about four, I think this is maybe my fourth or fifth event. So it's really great to be here. Thank you so much for inviting me. I also wanted to mention that actually Juvenescence sponsored a conference that Maria and I organised last year, the Young International Cell Senescence Association, cell senescence being an ageing kind of phenotype of cells. So yeah, we're also really grateful that we're here for Juvenescence as they supported us in our conference, which happened in this room. So yeah, this is the pointer. Cool, so my talk is about rapamycin, mTOR, and immunosenescence and DNA damage. So we'll get all into that. So when I started, I knew I wanted to do a project on immunosenescence because it is clearly one of the greatest challenges in the aging field. So what is immunosenescence? It's a really big, horrible word, but really it's just the senescence or aging of the immune system. So what that actually means is that if you look at older people, generally we see that their ability to mount an effective response to vaccines decreases, their resilience to novel infections, for example COVID-19, also decreases. And then we see an increase of cancer incidence. And then not only just getting the diseases, but the severity of the disease that you then subsequently suffer from is worse as we age. And when we actually go to look inside the blood of older people, we can see that there are some quite profound immune dysfunctions of those cells. So we have lots of different flavors of immune cells. I'm going to try and keep it not super duper immunologically technical, but we have different flavors of immune cells. And is that laser?
The pointer's on the bottom. No, not the bottom, the bottom.
Ah, that bottom, okay. So, when we look at different immune cell types, we can see that there are some dysfunctions. For example, B cells struggle to produce antibodies. T cells, which are a really important immune cell type that I'll be talking quite a lot about today, they also show dysfunctions such as decreased proliferation and an increase of DNA damage. And we're really interested in this DNA damage phenotype that we see in older people's immune cells, because there's actually now quite a lot of evidence that we synthesized in this review that we published during my PhD, that DNA damage in and of itself can actually drive immunosenescence and immune system aging. And so what follows, therefore, is that if you can find interventions that can protect against DNA damage, that these actually might have quite important therapeutic value in treating immune system aging. Because the problem with DNA damage is that it can drive a phenomenon that I mentioned just earlier called cellular senescence. So what is cellular senescence? I know we're quite a diverse audience today, so I'll just spend a bit of time talking about what it is and how we measure it. And Maria will also talk a little bit about this in her talk as well. So cell senescence is when we have a proliferating cell, a very healthy, happy proliferating cell. We'll have lots of those when we're young. But over time, they'll experience some kind of persistent stress, including something that drives DNA damage, like radiation, viral infection also induces DNA damage, or other kind of stresses. And this will lead to either cell death or cell senescence. And so cell senescence, as I'm showing here, they literally get bigger, at least if you do it in a dish. They have a huge amount of activation of the DNA damage response, which is measured by gamma H2AX, which I'll talk about in my talk, P53, and some other kind of metabolic dysfunction that I won't talk about. And also high expression of things that stop the cell from dividing. Because cells, when they receive a lot of insult, they want to repair that insult. And so senescence is kind of a mechanism to prevent kind of oncogenic transformation. If you have lots and lots of DNA damage, you don't want to keep dividing, you want to stop dividing and repair it, but then senescence also has some kind of downsides as well. And a couple of those markers that we see increase in senescence is this P21 and P16 as well. And the problem, as I mentioned, is that with aging, we see an accumulation in the burden of senescent cells in our tissues. And so my question, given this kind of phenotype of DNA damage in immune system aging, is, well, do these age-related immune cells actually show senescence? Kind of an obvious question. And so to answer this, we analysed human blood from people, from humans. Everything I show you today is going to be all in humans. And we analysed them using spectral flow cytometry, and in doing so, could identify many of the different flavours of immune cell types that I mentioned earlier. Doesn't matter what they are, I'm not going to talk about them individually, not of themselves, but these are just the different types that we can see in human blood. And what we then did was to look at the cells that we know expand with age. I'll call these age-related immune subsets. And then we measured all of these different markers of cell senescence. So, for example, this is the P21, the P53, P16, gamma-H2AX, that DNA damage marker, FSE, so cell size. I told you they get bigger, so do they get bigger? And also markers of mTOR activity. So just as a brief aside, so mTOR is essentially nutrient sensor in the cell that we know increases with cell senescence. That's why we also wanted to measure that here. And if we just look to the right of each heat map are the cells that we know expand the most with age. And what we saw was that, yes, these markers of cell senescence do seem to be enriched in these age-related immune cells across all of these different flavors of immune cell subtypes in the blood. Particularly, we saw that P21 and P53 were increased in aged immune cell types, and these are markers that we know are particularly enriched in DNA damage-induced senescence. And also that mTOR activity was increased, particularly in these T cells. And that was also the case with gamma-H2AX, so DNA damage. So that was very interesting to us. Of course, this is just looking at aged immune cells in a person and comparing them aged immune cells compared to the not aged immune cells within a person. What about if we take a group of people that are older and compare them to a group of people that are younger? And so that's what we did here. And this time I'm just going to show you the data for phospho 6, which is a marker of mTOR activity. And what we saw was that we compared these older people to younger controls and looked at all of these different immune cell subtype flavors. We do see an increase in mTOR activity as well. And this was really interesting to us because we know that low-dose mTOR inhibition improves vaccine responses in older people. So there have been a large number, a handful of very large-scale studies in older people where if you give them an mTOR inhibitor, give it 2 weeks of washout, and then perform some kind of immune challenge, so in these it was the flu vaccine, the mTOR inhibitor treated group had much higher antibody responses, T cell responses, that kind of thing. So we know that in the context of age, low dose mTOR inhibition improves immunity. But aside from knowing that mTOR inhibitors inhibit mTOR, we don't really know why that is. However, there are some really beautiful data in this paper that came out at the very beginning of my PhD that showed that if you look at a mouse, and you take a mouse and you engineer their immune cells not to be able to protect their DNA from damage, firstly, it really strongly phenocopies immunosenescence. So this is just, imagine giving a mouse a vaccine. That's what this is here. So in control mice, they are able to mount a response to that vaccine. But in these mice that have lots and lots of immune-specific DNA damage, they really can't mount that response. And this is very similar to what you see in an old mouse as well. But in this paper, they gave these mice rapamycin, and they saw that this did actually partially rescue their ability to produce an antigen-specific response. And so my question then was, well, does mTOR inhibition actually impact on human immune DNA damage and cell senescence? What's the relationship here? So to answer this was the bulk of my PhD, was developing this in vitro DNA damage assay where I take fresh whole human blood and then isolate the PBMCs, which are just the peripheral immune cells, activating them, then damaging them with a drug called zeaxanthin, which induces double-strand breaks, letting them recover, and then measuring their level of DNA damage response, this gamma-H2AX that I mentioned earlier. And we can see that, indeed, human T cells that have been exposed to zeaxanthin or, for example, hydrogen peroxide, do get damaged, so that's good, that's the model. So next what I asked was, that's just the quantification there. And not only do we see an increase in gamma-H2AX, but a whole variety of other markers of the DNA damage response. So they also increase, including P21 and P53, which you might remember from the age-related T cells and age-related immune cells. So my next question was to ask, well, what's the relationship between this DNA damage and this mTOR activity? So to answer this, we took the cells that we saw were stained very, very highly for DNA damage, so the cells that were very highly fluorescent for gamma-H2AX, and compared them to cells down here that weren't very damaged, and asked what's the difference in mTOR activity. And what we saw was that DNA damage was highly associated with having lots and lots of mTOR activation. So there is some kind of positive relationship here. but is it actually functional? So to answer whether they're actually related, this DNA damage and this mTOR activity, we performed the same assay of taking cells, damaging them, letting them recover, but this time did it in the presence of mTOR inhibitors. So rapamycin is a first generation, it is the mechanistic target of rapamycin is mTOR, so it inhibits mTOR. And then we also have AZD8055, which is a second generation rapalog, which also inhibits mTOR. And we asked, does it impact on this DNA damage response? So looking at gamma H2AX and then these other two markers of the DNA damage response. And what we saw was that, yes, as before, DNA damage with zeaxanthin led to high levels of damage and upregulating the DNA damage response, of course, but that mTOR inhibitors really seems to suppress this DNA damage response in T cells. And also they suppress levels of P21 and P53 as well. But of course, I'm just looking at markers of the DNA damage response. What about actual DNA lesions inside the nucleus? So to answer this, we performed a technique called the COMET assay. I hope it's obvious why it's called the COMET assay. Essentially, so as a side of what this assay measures, you take a cell and you just take out its DNA, and cells that have lots and lots and lots of DNA breaks, if you expose them to a potential difference, so a positive electrode, DNA, which is negatively charged, because it's all broken up and small, it will migrate really far. And so that is essentially your readout for whether a cell has lots of DNA damage. So in control cells, you'll just see a little dot, because there's not much DNA damage, it hasn't migrated very far. In cells that have got lots of DNA damage, you'll see that they've got this little tail, because all those DNA breaks have migrated towards that positive electrode. And the question is, what happens in rapamycin? Does it also protect against these DNA lesions measured by this COMET assay? And we can see that, yes, DNA lesions increase with zeaxanthin treatment, but rapamycin does decrease that. So DNA lesional burden is also lower with mTOR inhibition, not just the marks of the DNA damage response that I showed you before. And finally, we then looked at just cell fate, what happens to the cells after they've been exposed to DNA damage. And what we saw was that cells exposed to zeaxanthin alone, so the DNA damaging agent, generally were not well off. They died. But cells that had been exposed to zeaxanthin in the presence of rapamycin showed much greater survival by about threefold. And that's just quantified here. So this definitely shows some kind of enhancement of resilience against DNA damage with rapamycin and other rapalogs in vitro, but of course that was all in a dish with me in a cell culture hood. What about if we actually do this in people in the public? So that's what we did. So in collaboration with Phil Atherton and Dan Wilkinson at the University of Nottingham, who were performing this clinical trial, where essentially they took older people and treated them either with placebo or rapamycin, which is a low dose, of 1 milligram per day for a total of 4 months. And then we just took their blood at different points so I could analyse their immune system and see whether that was affected. And this is just to say that when we measured the blood rapamycin concentration, it was about 4 nanomolar, and all of the experiments I showed you before were done at about 10 nanomolar. So it is very physiologically achievable, the doses that we're getting in vivo as well. And so what we found firstly was if you just look at, and this is just T cells that I'm showing you here, if you look at the levels of mTOR activation and DNA damage, this correlates really strongly, ex vivo or in vivo if you like, suggesting that there is a relationship between mTOR activity and DNA damage in vivo. And that in rapamycin treated individuals, where we can see that they have lower levels of mTOR activity because we've inhibited it in vivo, they also have lower levels of DNA damage. And then when we look at P21, this cell cycle inhibitor that upregulates in cell senescence and DNA damage, this too was lower in rapamycin treated individuals across a variety of different cell types tested compared to placebo. So my take-home messages are that mTOR inhibition enhances resilience against DNA damage in vitro and in vivo, importantly. And this may be a new, therefore unrecognized role for mTOR inhibitors as genoprotectors. And so kind of model is that with age, we see an accumulation of DNA damaged cells that lead to organismal aging and immunosenescence, but that mTOR inhibition may enhance resilience against this and actually might be a very useful therapeutic value. And so this actually positions mTOR inhibition as that's me, as a kind of therapeutic strategy to target other pathologies that we know result from genome instability, including clinical radiation exposure, DNA repair deficiency syndromes like Werner syndrome, and even space travel. And I won't talk too much about this, but just to say that in our lab, in Garda's lab, they've recently set up the Space Innovation Lab to essentially test exactly this. So what happens if we take human tissue and put it in the International Space Station, essentially testing the hypothesis that space and the space environment might be a new model for accelerated aging. If you want to hear more about that, we've got a lot of people from Garda's lab sitting in the front row here who can wave. Talk to them. It's very interesting. Cool. So, thank you so much to my wonderful teams. My PhD was supervised, of course, by Lynn, but also Garda and Katya, and also we collaborated with the University of Nottingham. If you want to read the preprint, it's here. If you don't want to read the preprint, you can read the New Scientist article that also came out about our preprint, which is very cool. And of course, thank you so much to Jim and the Melon Longevity Scholarship, which without this, I literally couldn't have done any of this. So thank you. And yeah, love any questions.